Bismillahirrahmanirrahim. This is the third part of gram-negative non-enteric bacteria. And this is the last part of gram-negative non-enteric rods, which include francisella tularensis, or include zoonotic diseases that involve Brucella, francisella, Yersinia, and we will mention now francisella tularensis. The francisella tularensis accompanied with a specific infection called tularemia or rabbit fever, or others they call it as deer fly fever. This is a zoonotic disease transferred mainly from rabbits or from rodents, some types of rodents. This type of bacteria is relatively small in size, so they can be intracellularly parasite, can be intracellularly infect the host cells. So they are facultative intracellular. They can exist outside and inside host cells, especially in macrophages, the phagocytic cell, the tissue phagocytic cell. So this type of bacteria can invade the innate immunity, the immune system, by hiding itself inside macrophages. They are highly characterized by small in size, polymorphic, and they also has some specific factors or various factors like capsule, which is rich with lipids, not polysaccharides. It's lipid-rich capsule. The treatment of choice streptomycin, or we can use gentamicin. The third example is of zoonotic infectious gram-negative non-enteric bacillus, or we have another example called Pasteurella. Pasteurella usually refers to cucubacillus, short bacillus, exhibit very characteristic staining feature called bipolar staining. Bipolar staining. Bipolar staining means that not all the cell body stained wisely or fair staining. So some parts, or especially two parts, usually stained extensively, and the other parts are not stained. This is highly characterized the Pasteurella, and also Pasteurella are encapsulated in a specific capsule. The variance differs to this capsule and to other endotoxins, and usually the Pasteurella are facultative anaerobes. We can treat this type of infection because Pasteurella is a skin infection or wound infected organism. We can firstly treat the infection by cleansing and irrigating the wound. And in case of surgical or deep wound infection, we can use antibiotic, especially penicillin. The fourth example of zoonotic infectious gram-negative bacillus, Yersinia pestis. Actually, Yersinia, as we mentioned in the first lecture, in the first part of this lecture, refers to Enterobacteriaceae or enteric bacteria, because some features, or most of general features of enteric bacteria, involve or including the Yersinia pestis. Yersinia are gram-negative bacillus, facultative anaerobes, and also they lactose negative, oxidase negative. So most of features of Enterobacteriaceae exist in the Yersinia pestis. But why we have sometimes talking about Yersinia pestis and non-enterics? Because they refers to some non-enteric infections, like plague, for example, which is non-enteric disease. Plague is generalized systemic circulatory infection. Ta'un or plague or Black Death, one of the destructive epidemic diseases. And before human beings know about the antibiotic or explore the antibiotic of choice for Yersinia. It was one of the horrible disease, one of the very destructive disease, and killed, and from long time ago, killed about 25% of population in Europe, for example. Yersinia pestis can a leading cause for plague, and we consider it as zoonotic disease because the main reservoir is rodents, so we call it zoonotic disease. We can classify plague or Yersinia infection into three main groups. The urban plague, which transfer from rats, and sylvatic plague, which transfers from prairie dogs or from wild dogs, especially, and some rodents like squirrels in prairie and jungles, which is the most important reservoirs. And sometimes, unfortunately, some household pets like dogs, like cats, can be a reservoir for sylvatic plague. Actually, this type of infection, Yersinia pestis or plague, transferred through specific intermediate host or intermediate vector called fleas. Those fleas are sucking animals, sucking arthropods that sucks blood from dogs, from rats, and the infected blood usually involve the Yersinia pestis bacteria. And once this type of arthropods like fleas, when transferred from the animal to human and sucking the human, they inject the contaminated blood inside or through skin, through biting the human, and lead to infection. So the pathogenesis of this disease is transfer, disseminate the infection through lymphatic system and inoculation of lymph nodes and transfer to other parts. And the main characteristic of this infection, is local blood clotting and local hemorrhage under the skin, so it's characterized by black lesions under the skin due to the occlusion and due to the blood clotting, disseminated blood clotting in the whole the body. The treatment of choice is streptomycin, and we can treat the disseminated meningitis, disseminated infection like meningitis by chloramphenicol. There are several features of plague, and as we mentioned three types of plague: urban plague, sylvatic plague, and the third one is bubonic plague. And we can characterize bubonic plague, or we can indicate that bubonic plague is accompanied with pubis, the groin, lymph nodes inflammation, due to the aggregation of bacteria inside these lymph nodes, and it's highly characterized by bubonic, painful pubis. Those are the lymph nodes that exist in pubis. So we call it bubonic plague. The mortality is very high, about 50% of patients dying if untreated. The second type of plague is called pneumonic plague, and we can indicate the pneumonia. The infection occurs in lung. And finally, plague meningitis due to dissemination or ability of dissemination of organism to meninges and lead to meningitis. This is the main characters or the main features of blacking, hemorrhage under or beneath the skin in septicemic, for example, septicemic plague.